go ahead and get started. Thanks for being here this morning. I hope everyone had a fun night last night and saw a lot of great science yesterday. Uh, I'm Matt Kellinger. I'm the VP of Biochemistry at Element and one of the co-founders. And today I'm going to be talking about an application space using the AVITI system that's come out of a company that Element previously acquired earlier this year, Loop Genomics. This gives a long read option on the AVITI platform, and one of the things that attracted us to the technology is a use case in protein engineering. So we do a lot of protein engineering at Element. I'm a biochemist by training, and I've been working in protein engineering for 10 plus years. And surprisingly, at an NGS company, we still do a lot of Sanger sequencing for routine operations. And this is a really nice option that gets traditional Sanger methods onto an NGS system and allows for much greater throughput. So what's really provided by this method is protein engineering. There's multiple approaches. You can either do small scale or much larger scale with much higher throughput. And the challenge to the higher throughput model is how do you collect all the data? How do you get the genotypic data that connects with the phenotypic data that you're after? And whether it's engineering enzymes or synthetic biology or antibody engineering, oftentimes your best chance is to take a lot of shot on goals with a lot of clones. You need to get a lot of genotyping information on those clones. And if you go the Sanger route, you're sort of working in very parallelized fashion. That's also the challenge with current NGS approaches to Sanger replacement, is that you have to keep all of your clones, which you've isolated, separate throughout all of library prep because of how late indexing happens. And this solution, using the Loop technology, basically indexes upfront and first, then pools everything, and you run one library prep on thousands of clones instead of 1,000 concurrent library preps. And how this works is through some rather elegant molecular biology. But the first step of the process is to attach a barcode. In the case of the protein engineering or sequencing assumption, that is you start with a clonal well. So that's often the case for protein engineering applications. That barcode and the gene that it's attached to is then amplified several thousands of times. And then you run a distribution reaction, which acts as a molecular cut and paste, which effectively distributes that barcode within the longer read up to several kbs uniformly, so that you're able to identify the barcode inside the actual region of interest you're trying to sequence. Then perform some additional library prep and go into sequencing. Now you're sequencing your gene of interest, and the barcode gives you the location, so that informatically you can run de novo assembly on those buckets of data, put back together the entire long read sequence. The protocols are available online, the methods, as well as a host of publications. So one of the things about this technology is Loop's been around almost as long as Element has. They have about 25 publications through peer-reviewed journals that are up on their website. People using the technology for their applications, and I think that's important to look through to just show that there's a lot of use cases for this technology already in the field. So, I mentioned the workflow. A lot of times you start with automated colony picking. You're growing liquid suspensions of cultures in individual wells. Those can be sampled and placed on a library prep crate that adds barcodes unique to the well. And that gives you an identifier for what clone was in what well. After that step, as early as possible, you can basically pool all of those samples together and run one NGS library prep containing up to thousands of individual samples. And like I mentioned, the library prep is heavily PCR based inside a PCR machine. There's not much additional equipment that's needed to run over what we currently have for NGS applications. But you do those steps of indexing, the distribution reaction, and then ultimately sequencing. So to give you a use case for this data, previously when we looked closely at Loop, we ran a bunch of internal work on our own protein engineering projects to understand how well and how accurate it is. But there's customers in the world using this technology today on much larger scale protein engineering applications, and one of them is Ether Bio. So Ether Bio is a company that's really trying to build custom enzymes for real-world applications. They're taking a pretty novel approach here, where they're using a mix of AI, and they really need a lot of data to be able to understand how you create novel functions from enzymes. So it's a lot of computational data. It's also a lot of screening data. And with screening data, you need a lot of sequencing to be able to connect genotype to phenotype, both in the positive and in the negative case. So a lot of clones sequenced. And just to look again at how they're doing this. So a good plug for them. But really, they're doing large combinatorial libraries designed around generating novel activities from enzymes, right? This is a big task. And the more clones you can look at, the more success that is going to be had here. So just a brief look at some data. So what we've done is, on their plates that they're sequencing, there's a lot of controls, right? Controls are important for the protein engineering process throughout, and for cloning, it's also important. And one of the best sources of truth in the sequencing data for genotypes are the controls. So on each plate, there's several wild type controls, and on the right here, what you can see is just the alignment of some of those controls. And not surprisingly, that they're all the same. They're supposed to be, right? What we find in here is very, very, very high accuracy rates, right? So 14 errors total in almost 260,000 base pairs of sequencing is a very high accuracy rate. And then, of the 186 clones, 183 positively completely confirmed. So, one of the other challenges with Sanger, especially in protein engineering, is Sanger has dropouts around 5 to 10%. You have to go back, you have to repeat those clones. And that's a mess for a workflow because you've got to dive into last week or last week's month or last month's plates to be able to pull out those clones to resequence to get it. So we have a higher, overall full config build rate here. And then when you combine it with AVITI, you're really leveraging, the throughput of this system with, an easy library prep to get together to a relatively simple solution. And then to just look at, some of the numbers in here. So I wanted to include not just the controls, because they're usually not the fun thing to look at in protein engineering, but to look at the mutants. So you can see a distribution of mutants in there that are called. One of the nice things about really the Loop suite of technology is all of the bioinformatics are handled on the back end by them. So they build they take the short reads, they do the assembly, and in the end, you get a list of basically FASTQ files that have clone well addresses and full contigs already assembled. And the economics here are pretty good, as you would expect, using NGS versus using Sanger. Sanger's individual reads, we estimated $4 per Sanger reaction. If you have a gene where you have to tile several different reads, it gets quite expensive quite quickly. The advantage of LoopSeq and then the cost per G on AVITI really shines as the number of clones go up and up and up, because you're doing one library prep on many different clones, and then you're using a fraction of the sequencing power here in this case. So the savings for Ether was almost $16,000 just for these 4 384 well plates. So, briefly in conclusion, what this technology enables really is larger protein engineering projects, not having to make sacrifices based on throughput on the sequencing side. I think that early pooling is something that's unique and something that's empowering, because you're not spending a ton of reagent cost and time doing library prep. And then the cost advantages of AVITI really come together. Such a low cost per G. You can multiplex protein engineering work with this assay into other projects, use a little bit of the real estate inside other sequencing runs. That's a big advantage. And then, finally, I think the biggest one for me scientifically is the ability to access both positive and negative information. So a lot of times we're making trade-offs. We're only taking the positive hits for things. You're throwing away half your data if you're not taking the negative. And when you get all of that information together, you can better guide projects and better understand how function is being created. So a bunch of people to acknowledge in here, people on the Loop team, people on the Element team, and then the Ether Bio team. Protein engineering work takes many hands, and certainly that's a group of people that's worked hard on this one. So I can take any questions you have about